Hello everyone, my name is Sut Tun Naw and I'm a postdoctoral researcher at Sungkyunkwan University in Korea. So I recently graduated from the dual PhD degree program between NTU Singapore and SKKU in Korea. So I will be presenting what I did during this joint PhD program, as well as what I am doing after the program, which can be summarized into this title, which is the Lipid Bicelle Technology and Scientific Innovation and Research Life in Korea. Before I move on further, I would like to just give a little context as to how I came to Korea and then ended up working in Korea later. So I came to Korea as part of the requirement to fulfill the residency of this NTU-SKKU dual PhD program that is designed to promote research collaboration efforts between NTU and SKKU. So the students who are in this program need to complete the first half of their candidature at their home university and the second half at their partner university. So I finished my first half at NTU and I came to Korea for the second half, and I'm really enjoying my time here, making some new friends and also eating some delicious and new kinds of food besides doing research. And now I will talk about the research I did for PhD and the research that I'm still doing. So the focus of my research is on making supported lipid bilayers, SLBs, which can be used as modern membranes that mimic biological cell membranes and their key structural properties. So they are formed on solid supports and they are developed for fundamental membrane studies and applications such as biosensors and cell culture platforms, where they offer biological relevance and compatibility. And also, because these SLBs are formed on solid surfaces, they are very suitable for a wide range of surface measurement techniques. So there are a lot of techniques to fabricate SLB, and among them, vesicle fusion is the most popular. And in this vesicle fusion technique, the small spherical vesicles absorb and fuse on the solid support to form SLB. So the pros of this vesicle fusion are that it is fast and it is simple as far as the protocol is concerned, as all you need to do is just deposit the vesicles onto the substrate for the self-assembly to take place to form the SLB on the substrate. But the sample preparation is not that simple. The main challenge is to get the uniform size distribution among the small vesicles, and you need to have training and skills for that. And also, under the standard experimental conditions, the SLB formation is limited by many parameters. For example, if you add cholesterol in the vesicles, that will impede the SLB formation because cholesterol rigidifies the vesicles, so they cannot rupture completely to form SLB. So there is a great need to overcome some of these limitations, and an alternative SLB fabrication strategy has been tried out recently using Bicelles, and this method has proven very promising. So the Bicelles are made of two components, short and long chain lipids. So when these two lipids are mixed in an aqueous solution, the long chain lipids self-assemble into bilayers, and the short chain lipids form a rim around the edges of the bilayer. So this results in a disc-like structure. So, but this is just the generalized view of the Bicelle structure, and in fact, based on the Q ratio, which is the ratio of the long chain lipid concentration to short chain lipid concentration, and also other factors like temperature and composition, there can be many other different forms of Bicelle structures. So one main advantage is that the sample preparation is minimized since there is no need for specific equipment or wear control of the size distribution, which are the main challenges in vesicle preparation. And another advantage is related to the structure of Bicelles. SLB formation is energetically more favorable because Bicelles are usually larger and therefore have larger contact area than the spherical vesicles. So less adhesion energy is required for Bicelle absorption and subsequent SLB formation. So, but despite these advantages, the application of Bicelles in SLB fabrication is overall still very new. So because also there are also only a few studies that have reported on the use of these Bicelles for SLB fabrication. So a better understanding of the Bicelle-mediated SLB formation mechanism at the fundamental level is still needed, alongside the effects of the various experimental parameters, so that the optimal conditions for this SLB can be identified. And my research seeks to do exactly that, and in addition to move the Bicelle-based SLB fabrication beyond the lab. So these are the experimental methods I used. I use quartz crystal microbalance with dissipation, QCMD, to monitor the kinetics of Bicelle absorption and SLB formation by looking at the changes in the mass and viscoelastic properties of these adsorbed lipid layers, as indicated by the changes in frequency and energy dissipation signals. And besides these kinetic measurements, I used a fluorescence microscopy to visually observe the SLB formation process, and also did the fluorescence recovery after photo bleaching FREP experiments to characterize the lateral lipid mobility of the lipids, which is an important requirement in the SLBs. So before my thesis, our group has already identified this Bicelle-mediated SLB formation mechanism. As such, first, Bicelles in solution approach the substrate and then adsorb. After the Bicelles cover this entire surface, then they will fuse and form the SLB as a result of this short chain lipid returning to the bulk solution, because they have a higher solubility than the long chain lipids that remained on the substrate to form the SLB. So the entire process is captured in QCMD and this fluorescent microscopy as such. So note that SLB values are around, around minus 25 in QCMD, around minus 25 Hertz for frequency, and around 1 or less than 0.5 if it is very good quality for dissipation. So these are denoted by the dotted lines in the QCMD graph you will see later. So first off, I studied the effect of surface charge of both Bicelle membrane and substrate on SLB formation. So I tuned the Bicelle charge by adding positively charged lipids, DOPC, and negatively charged lipids, DOPS, in various molar fractions. And for the substrate type, I used the standard substrate SiO2 as the control, which is a negative substrate, and TiO2 as a negative substrate, and aluminum oxide as a positive substrate. So the results are as expected according to the electrostatic interactions, which is that the positively charged Bicelles form SLB on negatively charged substrates, and negatively charged Bicelles form SLB on positively charged substrates. So even though this trend is like expected, the significance here is that the amount of charged lipids required to form SLB on these substrates is much less than that required by the Vesicle fusion method. Next, I checked the effects of NaCl concentration on SLB formation from Bicelles and versus from vesicles. So the test range was from 0 to 500 millimolar NaCl. In the case of Bicelles, we saw that concentration does not affect SLB formation. SLB formed in all ionic strength conditions. And by contrast, in the case of vesicles, SLB form only from 50 millimolar NaCl onwards. But also they have slightly higher shifts than normal. So an SLB formed from Bicelles has higher quality, as shown by the uniform fluorescence, than SLB from vesicles, where there are some unruptured vesicles, which are shown by these bright spots. So these results prove that Bicelle method is much more robust than Vesicle fusion method under different ionic strength conditions. Next, I checked the SLB formation from Bicelles that contain like up to 60% more cholesterol. So with the conventional Vesicle fusion method, vesicles that have more than 20% more cholesterol cannot form SLB. But with vesicles, I found that as complete SLB can be formed up to 30 more% cholesterol, and incomplete SLBs at 40 to 60 more% cholesterol, meaning that there are some bicelles in the SLBs. But and then this is confirmed by the FREP results that show the reduction in the lipid mobility at 40 to 60 more% cholesterol. So these incomplete SLBs can still be useful for biophysical studies because bicelles still show rupture here, and then so that therefore they have planar bilayer regions in these bicelle layers. And so the next part of my research is to explore how this Bicelle technology can be moved beyond the lab scale fabrication. So to do that, I look for suitable replacement for DHPC, which is the synthetic and very expensive lipid. So to be able to upscale the SLB fabrication beyond the lab, a natural and cheap alternative molecule is desired. And accordingly, I chose the natural and cheap detergent molecule called Lauric Acid, which is LA, which is used in soap making. And the results show that SLB formation at low LA amount, and as well and considering that SLB LA is more than like 6,000 times cheaper than DHPC, it is very suitable for low cost and large scale SLB fabrication beyond the lab. So in conclusion, my PhD research has produced the following new findings: that the Bicelle surface charge can be fine-tuned to fabricate SLB on different oxide surfaces with different charges, and that SLB can be formed from Bicelles regardless of salt concentration in the bark, and that cholesterol-rich Bicelles can form cholesterol-enriched SLBs. So all of these have shown the robustness and competitive advantage of the Bicelle method, and in addition, I have also demonstrated that the expensive DHPC lipid can be replaced with the natural and cheap compounds such as lauric acid to move the SLB fabrication capability beyond the lab. So that was what I did for my PhD, and now I would like to talk very briefly about the research I'm doing here as a postdoc. So basically, I'm continuing to work on lipid-related research, and I will explore a bit more applications related to biosensor platform development. Besides research, and I'm learning some Korean language so that I can communicate here, and also exploring some different places in Korea to experience culture and stuff. So overall, I'm quite enjoying both research and life in Korea. So this is the end of my presentation. Thank you for listening and enjoy the rest of the conference.